the cell cycle and cell division. In order to understand how we make more cells, we need to understand every part of the cell cycle. When you are dividing cells in your body, you will do this to grow further, to repair a certain amount of area, or to develop, to be able to differentiate your cells by making more cells, accessing different genes. The cell cycle has 4 major phases. The first 3 of them are going to fall into a larger phase we call interphase. Interphase consists of G1, S, and G2, and encompasses everything that happens within those. During G1, the cell is going to grow in order to have enough volume to make 2 cells, and it's going to synthesize more organelles. It's going to make a second set of Golgi apparatus. It's going to make more ribosomes. It's going to make more mitochondria. Then in the S phase, DNA synthesis will occur, in which case we'll take our chromosome, or we'll do it while it's in a loose, uncondensed chromatin form, and we'll duplicate it, making the exact same copy of DNA a second time. And then in G2, we will do our final amount of growth, mainly prepping for mitosis. Mitosis, or M phase, will be the final phase. In mitosis, this is going to be occurring in somatic cells. Somatic just means body. So your skin cells, your blood cells, your bone cells, all of that is where you will see mitosis occurring. And it results in 2 identical daughter cells that are going to be diploid. Diploid means that it's going to have 23 pairs of chromosomes for a 46 chromosome number. So it's going to be ready to have all the genetic information you need for each of your cells. PMAT is how we can remember prophase, metaphase, anaphase, telophase, in which case those are the 4 steps of mitosis. The whole goal of mitosis is to interact with the nucleus and the chromosomes in order to properly get an even amount of DNA on either side. When we are looking at our chromosomes, there are 3 structures that you need to know. The centromere is going to be the center part that holds it together. A chromatid is going to be a single leg of the chromosome. And sister chromatids are going to be identical sections of chromosomes that are right next to each other. So, in prophase, we're going to dissolve the nucleus. We're going to break down that nuclear envelope, thus revealing our chromosomes from here on out. And then we'll begin to form centrioles and we'll begin to form spindle fibers. In metaphase, those centrioles will have moved to either end of the cell, and then our spindle fibers are going to attach to the chromosomes, lining them up along the middle. And then in anaphase, those spindle fibers are going to shorten and pull apart the chromosomes. More specifically, we're pulling apart the sister chromatids from each other. And it's because of this that this piece of information is identical to here, identical to here, identical to here. So therefore, in telophase, when we reform the nucleus around our genetic information, these are identical halves of the cell. And in cytokinesis, when we cut them into 2 cells, we get 2 genetically identical diploid cells. That is mitosis. That is somatic cell division. We constantly need to make sure that this is not occurring uncontrolled. Uncontrolled or unregulated cell division is cancer. It is our inability to control cell division. The way we normally control cancer or unregulated cell growth is through 3 checkpoints, G1, G2, and M checkpoint, and with the help of CDKs or cyclin-dependent kinases and cyclins. CDKs exist and they will be bound to by a cyclin, this blue box here. The cyclin will activate the cyclin-dependent kinase, and what will happen is, at each checkpoint, we will look for things such as growth factors. Do we have room to expand our skin here? Do we have enough nutrients to continue growing? If the answer is yes, we'll continue on. If the answer is no, we'll go into the G0 phase, or cell arrest. Then we'll continue to get to the G2 checkpoint. Did we properly replicate our DNA? If the answer is yes, we'll allow it to continue. If the answer is no, then our CDKs bound with our cyclins will induce apoptosis, which is programmed cell death. It's cell suicide. We're going to tell this cell to kill itself because of the fact that DNA did not replicate properly, and it's going to be cancerous. That is cancer. Meiosis is going to be sex cell division. This only occurs in spermatogenesis or oogenesis, meaning that we're making gametes, we're making sex cells. This is going to result in 4 unique haploid cells. There are 2 marquee differences. The first of which is in prophase 1, crossing over will occur. And what's going to happen is our chromosomes are going to overlap and recombine sections of DNA, making it genetically unique from here on out. Then we'll go through metaphase 1, very similar. We're going to line up our homologous chromosomes this time. Anaphase 1, we're separating entire chromosomes, not chromatids. Key difference. Telophase 1 will reform. No interphase. After this, we'll go right into prophase 2. Same thing occurs. Metaphase 2, anaphase 2. We're going to separate our chromatids now. And then telophase 2, we end with our 4 genetically unique haploid cells. Good luck studying. I'll see you guys all soon.